Welcome back friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding CpG islands. Is it clear? What are CpG islands? Where the CpG islands they are available in a double-stranded genomic sequence, and what are the various consequences which are in association with the CpG island which is available in a double-stranded genomic sequence in detail. Is it clear? So here, prior to the explanation of CpG islands in detail, so here what is CpG? Is it clear? So let me write it on the board. So what it is? CpG islands. CpG. Is it clear? So here C and G, likewise the A and T, these are the specialized dinucleotides which are available in a particular double-stranded genomic sequence. Then why they are termed as CpG islands? So here, though the specific CG repeats, they are available throughout the genomic sequence, but at some particular site of the double-stranded genomic sequence. So here these specific CG repeats, they are available at higher proportion. Is it clear? So here, due to the availability of the specific CG repeats at higher proportion at some particular site of the double-stranded genomic sequence, so here they are termed as what? CpG islands. Is it clear? So for your better understanding, let me print it on the board. So here, let us consider one double-stranded genomic sequence. And here it is the other complementarity strand, right? Is it clear? So here all these are the complementarity nucleotides. A, T, G, C, again it is G, again it is C, G, C, G, A, T, A and A. Is it clear? And here all these are the complementarity nucleotides. So here the complementarity of A is T, A, C, G, C, G, C, G, C, T, A, T, T. Is it clear? So here it is nothing but what? Double-stranded genomic sequence. So here, though the specific CG repeats, they are available throughout the genomic sequence, but at some particular site of the double-stranded genomic sequence, here the specific CG repeats, they are available at higher proportion. Is it clear? So here, due to the availability of the specific CG repeats at higher proportion, here they are termed as what? CpG islands. So here CG, these are the common nucleotides which are a part of the double-stranded genomic sequence, and whereas here P is nothing but what? The linking phosphate. Is it clear? So here, what is the uniqueness of the specific CpG island here? So here if we consider the cytosine nucleotide which is a part of the CpG island. So here in general, when the specific cytosine nucleotide, when it undergoes deamination or when it loses its amino group, it induces uracil. Is it clear? So let me print it on the board. So here if we consider the cytosine nucleotide, so here it is nothing but what? The cytosine nucleotide, right? So here, when the cytosine nucleotide, when it loses its amino group, so here it induces uracil. Uracil, right? So here it is nothing but what? The cytosine nucleotide. And here it is nothing but the uracil. Is it clear? So in general, if we consider in case of the double stranded genomic sequence, there is an availability of cytosine nucleotide. So here once when the specific cytosine nucleotide, when it undergoes deamination, it induces uracil. But here in case of the evolution point of view, in case of the vertebrate DNA, instead of having the normal cytosine nucleotide, here the specific cytosine nucleotide, it is successfully modified into 5-methyl cytosine. Is it clear? So here in general, when the specific cytosine nucleotide, when it undergoes deamination, it induces uracil. But here, instead of having the normal cytosine nucleotide in case of the vertebrate DNA, when the specific 5-methyl cytosine, when it undergoes deamination, under such condition, here it induces thymine nucleotide. Is it clear? So let me print it on the board. So here, let us we consider it as the 5-methyl cytosine. 5-methyl cytosine, right? Five-methyl cytosine. So here it is nothing but what? 5-methyl cytosine. So here once when the 5-methyl cytosine, when it undergoes deamination or once when it loses its amino group, under such condition, here it induces the thymine nucleotide. So here it induces the thymine nucleotide, right? So here it is nothing but the thymine nucleotide. Is it clear? So here in general, when the specific cytosine nucleotide, when it undergoes deamination, it induces uracil. But here in case of the evolution point of view, in case of the vertebrate DNA, the normal cytosine nucleotide, it is successfully modified into 5-methyl cytosine. So here, once when the specific 5-methyl cytosine, when it undergoes deamination, under such condition, here there is an availability of thymine nucleotide. So here, due to the availability of the thymine nucleotide during the deamination, it may cause the transition mutation. Is it clear? So here, in order to overcome the problem, the specific mechanism, the specific process, it takes the advantage of a specific covalent modification, which is nothing but methylation. Is it clear? So let me write it on the board. What it is? Methylation. Methylation. So here what exactly happens during the methylation? As the name itself suggests, during the methylation, successful recruitment of methyl group takes place. So here, in order to recruit the methyl group to the double-stranded genomic sequence, here there is a requirement of a specific enzyme which is nothing but DNA methylase. Is it clear? So here by taking the advantage of DNA methylase, these specific methylases, they are very specific. Is it clear? Here these specific DNA methylases, they are very sequence specific and as well as base specific. So here by taking the advantage of DNA methylases, the specific methyl group, it successfully recruits to the modified cytosine nucleotide. Under such condition, the modified cytosine, it won't undergo deamination. Is it clear? So here we need to understand one of the important strategy. Though the methylation, it is capable to protect the specific modified cytosine nucleotide from undergoing oxidative deamination, but in addition, the specific methylation, it is capable to inhibit the gene expression. Is it clear? So here methylation, it is the specific strategy through which the gene expression, it is successfully inhibited in case of the various cells. So here the gene expression, it successfully inhibits in the presence of the specific methyl group. Is it clear? So here, as I've already mentioned you, the specific CpG islands, they are available throughout the genomic sequence. So here, if the specific CpG island, if it is available in the rest of the genomic sequence under such condition, if the specific modified cytosine nucleotide, if it undergoes methylation, it won't cause any major issue. Is it clear? So if the specific CpG island, so here let us consider it as the CpG island, right? So here if the specific CpG island, if it is available in the rest of the genomic sequence, under such condition, if the modified cytosine nucleotide, if it undergoes methylation, it won't cause a major issue. But here the specific CpG islands, they are available even at the specialized sites which are responsible for initiating the transcription mechanism, and they are nothing but what? The promoter region. Is it clear? So let me write it on the board. CpG islands are present within promoter region. So here, if the specific CpG island, if it undergoes methylation in the rest of the genomic sequence, it won't cause a major issue. But here, if the specific CpG island, which is present in case of the promoter region, if it undergoes methylation, under such condition, the gene expression, it won't take place. Is it clear? Because as we all know, the specific methylation, it is the specific strategy through which the gene silencing, it is most commonly organized in case of the various cells. So here, in order to overcome the problem, is it clear? In order to overcome the problem, the modified cytosine nucleotide, in case of the promoter region, it is successfully replaced by a specific cytosine analog, which is nothing but 5-azacitidine. Is it clear? So let me write it on the board. What it is? 5-azacitidine. So here, what is 5-azacitidine here? The specific 5-azacitidine, it is an analog of cytosine. And even it is an inhibitor of DNA methylase activity. So let me write it here. 5-azacitidine, it is an inhibitor of DNA methylase activity. Is it clear? If the normal specific cytosine nucleotide or the modified form of the cytosine nucleotide, if it is removed and if it is replaced by 5-azacitidine, under such condition, the specific methylase enzyme, it is unable to recruit the methyl group to the specific 5-azacitidine. So under such condition, the specific RNA polymerase enzyme, it is capable to recognize the promoter region, and even it is capable to organize the gene expression so accurately. Is it clear? So here, the specific CpG islands, which are available throughout the genomic sequence, they play an important role in case of the epigenetic modifications. Is it clear? So here, what are the various advantages of the specific CpG islands which are available in a particular double-stranded genomic sequence? So here, most commonly, the specific CpG islands, they are responsible for inducing the gene silencing mechanism. Is it clear? So let me write it on the board. So let me arrange the things here. CpG islands, they are responsible for inducing the gene silencing. Are responsible for gene silencing. Is it clear? And even in order to regulate the genome imprinting, the specific CpG island, it plays an important role. Is it clear? Genome imprinting, it successfully regulates because of the presence of the CpG island. In addition, the specific CpG islands, they are most commonly responsible for inhibiting the gene expression of various intragenic parasites and as well as the various carcinogenic viruses which successfully enter into an eukaryotic host cell. Is it clear? So here, due to the presence of the CpG island within the promoter region, so once when the specific CpG island, when it undergoes methylation under such condition, so here the gene expression, it successfully inhibits in case of the eukaryotic host cell. Is it clear? So here this is regarding the CpG island, which is a part of the double stranded genomic sequence which is most commonly observed in case of an eukaryotic host cell. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.